Human skin cells can be reprogrammed into brain tissue, growing millions of neurons that fire electrical signals identical to premature infant brain waves. These organoids spend their short lives in laboratories as test subjects for toxins, diseases, and drugs that researchers need to evaluate safely. At UC San Diego, organoids are mounted on robot platforms and navigated through physical mazes to test their learning and decision-making capabilities. Melbourne startup Cortical Labs integrated living neurons into a biocomputing system, where the cells learn to play Pong by receiving electrical rewards and punishments. Alison Muatri, a Brazilian developmental biologist at UXT, grew organoids from autistic donors, including his own son, to understand where neural development diverges. Scientists create induced pluripotent stem cells by treating adult skin cells with specific proteins that revert them to an embryonic state. Brain organoids remain opaque and snot-colored, roughly the size of a chia seed, containing about 5 million cells, including 5 million neurons. Federal law exempts invertebrate research from animal welfare regulations, so organoids are not legally classified as animals requiring special protections. Without electrical stimulation or sensory input, organoids exist in isolation, raising philosophical questions about whether consciousness could develop in such conditions. Researchers apply electrical pulses and chemical signals to organoid cultures, observing that the living tissue responds, remembers, and eventually anticipates patterns. Cortical Labs operates a grid of 59 electrodes, each supporting a tiny culture of human neurons that can communicate across 8,000 miles. The biological computers, called CL1, resemble elongated toasters and maintain life support systems, keeping up to 1 million neurons alive for 6 months. In 2022, Brett Kagan trained a neural culture on a microchip to play Pong by rewarding correct decisions with predictable electrical pulses. The neurons reorganized to avoid chaotic electrical bursts, demonstrating that living biological systems minimize surprise and can be programmed through environmental feedback. Some researchers claim the organoid playing Pong displayed a form of sentience, causing significant backlash from the neuroscience community over misusing the term. Critics note that consciousness requires embodiment and environmental interaction, but organoids in dishes receive no sensory feedback from the physical world around them. Annie Cathuria at Johns Hopkins points out that consciousness remains qualitatively defined and impossible to measure objectively in tissue floating in a dish. Philosophers debate whether organoids could become trapped islands of awareness inside their dishes, raising profound ethical concerns about creating suffering beings. Jonathan Birch argues that a brainstem would function as the power cable for consciousness, making larger organoids with complete neural structures morally significant. Organoids naturally stop growing at 5 millimeters because they develop necrotic cores and suffocate without a vascular system delivering oxygen and removing waste. David Gracias at Johns Hopkins engineers biomimetic artificial arteries to overcome the oxygen limitation and allow organoids to grow much larger. Lena Smirnova grows rough blood vessel networks from endothelial organoids, advancing toward the goal of sustaining larger, more complex brain tissue cultures. Alison Muatri's longevity record for organoid survival is 3 years, until someone accidentally dropped the dish, demonstrating the fragility of this research. Thomas Hardung advocates for 1 centimeter organoids at mouse brain scale, requiring perfusion systems to solve the current bloodlessness problem limiting growth. In 2023, the international scientific community published a declaration christening the field organoid intelligence, generating significant press coverage and funding interest. Current funding focuses on drug testing for neuropsychiatric conditions, since 95% of drugs fail in human clinical trials after animal testing. Animal testing uses non-human primates with electrodes implanted in their brains, typically only 5 or 6 subjects per experiment due to ethical constraints. A single 6-well plate can produce up to 4,000 organoids, dramatically increasing research capacity compared to traditional animal model limitations. Organoid intelligence research serves as an instrument for studying learning, memory, and drug effects, rather than as a final destination for understanding consciousness. The NIH announced in July 2025 that it would cease funding research relying exclusively on animal models, promoting organoid alternatives instead. The NIH allocated $87 million in September 2025 to establish a standardized organoid modeling center for coordinated research development. Organoids derived from consenting adult donors avoid religious backlash that targeted embryonic stem cell research and don't trigger animal rights activism concerns. Public opinion surveys indicate most people feel minimal moral objection to organoids, with discomfort stemming from the creepy sensation of blurred boundaries. In 2017, neuroscientists created a brain organoid from journalist Philip Ball's skin cells, which pulsed with electrical signals matching human brain activity. Ball described feeling unsettled when viewing his own neural tissue under a microscope, recognizing that neurons were communicating despite lacking conscious thought. In his book How to Grow a Human, Ball explains why rewinding cellular development dissolves the coherent unity and continuity of individual identity. Life develops linearly from fertilized egg to zygote to trillions of cells, but extracted cells abandon that singular history and restart independently. The question of whether extracted neural cells still belong to the original person remains philosophically unresolved and legally ambiguous across jurisdictions. Ted Chiang warned that creating conscious biological agents would inevitably produce billions of entities capable of suffering and cause immeasurable harm. The AI industry assumes machine intelligence inevitably surpasses biological intelligence through silicon and neural networks at enormous financial and environmental cost. Biologists emphasize that consciousness remains profoundly complex and resist calling organoid behavior sentience without rigorous biological proof and clear ethical frameworks. Silicon-based computing models the brain through top-down neural network architecture, while biological systems adapt bottom-up through evolutionary processes far more efficient. Truly adaptable computing systems require flexible substrates that can self-repair and evolve, characteristics silicon cannot provide but biological neurons possess naturally. Throughout evolutionary history, only biological systems have ever produced genuine intelligence through adaptation, self-organization, and environmental interaction across millions of years. The axons in Ball's library exhibit stretch toward each other across empty space, the same impulse driving human connection since birth. Whether silicon or living neurons dominate future computing, human cells will continue their fundamental drive to connect and communicate regardless of technological progress. The future of intelligence might not be artificial at all, but rather a partnership between biological neurons and technological systems working in harmony. These organoid studies represent humanity's attempt to understand the fundamental nature of mind by building it from scratch in controlled laboratory conditions. The implications extend beyond medicine and computing into philosophy, ethics, and existential questions about the nature of consciousness itself. What begins as basic research into neurological disease may ultimately reshape how humanity creates, understands, and relates to intelligence itself. The living neurons growing in laboratory dishes today represent the bridge between classical biology and a future where consciousness itself becomes engineerable. This research demands we confront uncomfortable truths about the nature of mind, suffering, identity, and what consciousness truly means at fundamental levels. Don't forget to subscribe and turn on notifications to stay informed of the latest videos.